Hello students, this is Mrs. Shweta Laddha from Fergusson College, Pune. And if you remember, we are discussing about our first chapter, reproduction in lower and higher plants. Last time, if you remember, we had discussed about structure of pollen grain. After that, we also discussed where the pollen grain is formed in the anther, in the pollen sac. We also in detail studied about the TS of anther, the different layers which are present in the anther, microsporogenesis, how it is occurring, and afterwards what are the different changes that occur in the pollen grain when it is going to develop into a male gametophyte. And these changes we discussed in two steps. The first step is when it is still inside the pollen sac, how it forms a two-cell stage, having a small generative cell and a larger vegetative or tube cell. The generative cell, it will produce the two male gametes, and the vegetative cell, it will liberate. Then this two-cell stage, it gets deposited on the stigma of the flower. Because of absorption of sugary nectar, it swells up, osmosis, it swells up, endosmosis occurs, and there is pressure on the intine and a pollen tube is formed. This pollen tube, it starts traveling through the style to reach the ovule because of chemotactic movement. These are important words. And then degeneration of the tube nucleus occurs, whereas the two male gametes are going to play very important role in double fertilization in angiosperms. But now the ovule is not yet ready. So in today's lecture, I am going to teach you structure of Anatropus ovule or structure of ovule, different types of ovule, and how the fertilization will occur. So let us begin with our PowerPoint presentation. Yeah, students. So let us start with the structure of Anatropus ovule, which is the most common ovule present in angiosperms. Structure of Anatropus ovule, if you look at this picture, there are different parts to a inverted Anatropus ovule. It is attached to the placenta by a stalk, and this stalk, it is called as funicle. I'll just give you a brief picture. Where is this present? So this is our stigma, this is our style, this is our ovary. Inside the ovary, this is the funicle, the stalk, and to which the Anatropus ovule is attached. So now it will become easy for you to understand the structure of this ovule. So this stalk, and what is there at the bottom? There is placenta. What is placenta? It is the nutritive layer which is going to provide nourishment to our developing ovule. So it is attached, the ovule is attached to the placenta with the help of a stalk. And what is this stalk called as? Funicle or funiculus. The point of attachment of funicle to the body of the ovule is called as hilum. Look at this. This point of attachment, it is hilum. This is hilum. Funicle, it forms a ridge on the body which is called as raphe. Look at this raphe. This black color strand which is present. This is raphe. And basically raphe, it consists of the conducting strand which is going to conduct water, mineral. So raphe is there which is arising from the placenta, going through the funiculus, the hilum, and it goes to the body of the ovule. Ovule contains nutritive parenchymatous tissue which is called as nucellus. Look at this labeling, nucellus. I am just highlighting the nucellus. This is the nucellus. And nucellus, it consists of what type of tissue? Parenchymatous tissue. And what is their function? They are nutritive in function. Nucellus is protected by two multicellular coats, two integuments. And as there are two integuments, such ovule, it is called as bitegmic. Look at this. This is the outer integument and this is the second inner integument. Outer, inner. As there are two integuments, this ovule is called as bitegmic. Basal portion of the nucellus from where the integuments arise is called as chalaza. This is the basal portion. From here the integuments are arising. So this is called as chalaza. Names are difficult. Read it again and again. You will learn them by heart. And I will suggest you draw this diagram with me. Small opening present at the apex of the integument is called as micropyle. This opening which is present at the tip, it is called as micropyle. I repeat, funicle, raphe, stalk, where the point where the funicle is attaching to the body, hilum. Then this base is chalaza. From chalaza, what is arising? Two integuments, so bitegmic. Nutritive parenchymatous layer is called as the nucellus, and this opening at the other end is micropyle. Female gametophyte or embryo sac is present in the nucellus. Rather, one of the cell of the nucellus, it gives rise to form this embryo sac, which is present inside the nucellus. I hope you followed basic structure. Let us proceed. There are different types of ovules. 6 types of ovule we will discuss. I taught you about the structure of Anatropus ovule, also called as inverted ovule. But angiosperms may show presence of different shapes of ovules. If it is an everted ovule, it is called as orthotropous. Look at this, micropyle is at the top. Okay, this is the chalaza towards the funicle. This is called as the orthotropous ovule. Now, if you look at the Hemi Anatropus is half. Hemi is half. Ana is inverted, but it is not completely inverted. It is at right angle. So, here you can see the micropyle and the chalaza. They are at 90 degree to the funicle. This is Hemi Anatropis. Camelotropus, if you look properly, the nucellus becomes little bit, what we'll say, curved. Okay, and the micropyle is towards the base, which is towards the funicle. Such type of ovule is called as camelotropous ovule. In amphitropous ovule, you will see the nucellus becomes horseshoe shaped. If the nucellus is becoming horseshoe shaped, micropyle is coming towards the funicle, it is called as amphitropous. And last, you see that if the ovule is taking a 360 degree turn, then we call it as a circinotropous ovule. Complete turn 360 degree, it is called as circinotropous ovule. So these are the different types of ovule, but the most common type of ovule is Anatropus ovule. You have this is in your practical also. So you just revise the structure of Anatropus ovule. A 3-mark question can be asked to draw and explain the structure of Anatropus ovule. They may give you the diagram. They may ask you to tell the function or identify the parts of the Anatropus ovules. Let us proceed now. Let us see how the development of female gametophyte, that is Megasporogenesis, takes place. Development of male gamete was called as Microsporogenesis. Similarly, development of female gamete, it is called as Megasporogenesis. I have taken this diagram from your textbook so that it becomes easy for you to understand. Let us see this step by step. A diploid hypodermal cell at the micropylar end of the nucellus gets differentiated to form archisporium. Let me again explain you. So I am saying about hypodermal, hypo below the epidermis, okay, towards the micropyle. Do you remember this is the Anatropus ovule, this is the bitegmic, okay, so this is the micropyle, okay, this is the chalazal end, the nucellus. What is they saying? This is the micropyle, this is the funicle. Single cell at the micropylar end, towards this end, one cell towards the micropylar end of the nucellus, it starts forming archisporium. This single cell or archisporium, it functions as megaspore mother cell. See, there in the male gametophyte we had called it as microspore mother cell, which gave rise to 4 microspores haploid. Here we are calling it as megaspore mother cell. Okay, this megaspore mother cell undergoes meiosis to form tetrad of haploid megaspore. So this is that megaspore mother cell which is diploid. It undergoes meiosis and it forms a tetrad. Tetrad means 4 haploid megaspores. So these are 4 haploid megaspores linearly arranged in a straight line. This process of formation of 4 haploid megaspores from the diploid megaspore mother cell, it is called as Megasporogenesis. Here also 4 are formed, but 3 degenerates. Megaspores are generally arranged in a linear tetrad. They are arranged in a straight line. Generally, the chalazal megaspore remains functional. Chalazal means towards the top. So, this diagram is ulta. Here, this side is the micropyle and this side is the chalaza. Okay? No issues. You try to understand which one remains. The one which is towards the chalazal end remains and other 3 degenerates. The one which are towards the micropyle, they degenerate, and the one which is towards the chalazal end, it remains. So, students, please remember here, in megasporogenesis, 1 MMC finally give rise to only 1 megaspore, haploid megaspore, correct? But in microspore formation, 1 microspore mother cell gave rise to 4 haploid microspores. Here it gives rise to 1 megaspore. Which one is remaining alive? The one which is towards the chalazal end. What happens to the remaining 3? They get degenerated. Now further what happens? This one is remaining alive, it undergoes growth, it grows, and once it is growing by nourishment, it becomes enlarges in size. Okay, then it starts undergoing mitotic division. When it undergoes mitotic division, it is initially haploid, isn't it? It is haploid. When it undergoes mitotic division, initially only nucleus divides, the cytoplasmic division does not occur. After one mitotic division, you will give rise to two nucleated, not two cell, I'm sorry, two nucleus are formed in one cell. And then after second mitotic, you will see this nucleus give rise to more two and this nucleus give rise to more two. So four nuclei are formed. After this, again it undergoes mitosis and from 4, 8 are formed. I'll draw here. So these two will form 4 at the top, 1, 2, 3, 4 at the top, and this will form 4 at the bottom, correct? So there are 8. Out of 8, one moves toward the center from this end, one moves from the center towards this end. So in the center there are 2 nuclei, 3 remains at the top, 3 remains at the bottom, correct? Now the one which are remaining, which are at the top, they form the antipodal cells, and the one which are at the bottom, one forms the egg, the one in the center forms the egg, and the one on the either side, they form the antipodal. So these are two synergids. They form the synergids. In the center is the egg, and on the sides the two are forming the synergids. Okay, and in the center there are two female gametes which fuse together to form the secondary nucleus. Let us proceed. We'll study this process in detail. So functional fertile megaspore is the first cell of the female gametophyte. It enlarges. Haploid nucleus of the functional megaspore undergoes 3 successive free nuclear mitotic division. How many nuclear mitotic division? 3. So initially it is haploid having single nucleus. After first mitotic division, it will give rise to 2 nucleus. After second, it will give rise to 2 nucleus. Then after this, it will give rise to 4 nucleus. 4 at the top. Okay, this will give rise to 8 nucleus. So in this way, there it gives rise to 3 mitotic divisions. First mitotic results in the formation of 2 nuclei. We saw formation of 2 nuclei. Both nuclei again undergoes 2 successive division forming 4 nuclei. So see, 1, 2, 3, 4, 4 nuclei are formed, and finally 8 nuclei are formed. So here 4 at the top and 4 at the bottom. So 8 nuclei are formed. So now there are 4 nuclei at each pole. One nucleus from each pole comes to the center of the, and they function as a polar nuclei. This is followed by cellular organization to form 3-celled egg apparatus at the micropylar end. Micropylar end at the bottom, towards the micropyle, 3-cell stage. Center, it has egg, and on the side there are 2 haploid cells which act as synergids. Together, these 3 cells at the micropylar end, it is called as egg apparatus. And there are 3 antipodals at the chalazal end. This is the chalazal end. Towards the chalazal end, the 3 haploid cells, they are called as the antipodals. And 2 which remain in the center, they form polar nuclei. Okay. Later, these polar nuclei will fuse together to form a secondary nucleus, and it is formed by fusion of 2 haploids, so it is diploid in nature. Thus, 8 nucleated, 7-celled female gametophyte is formed within the functional megaspore. I repeat, how it is 8 nucleated? 1, 2, 3, 3 antipodals at the chalazal end, 4th egg, 5th and 6th synergid, and this entire one is 7. So 7-celled. How many nucleus? 8. Why 8? 3 at the top, 3 at the bottom, and 2 in the center, fusing together later to form secondary nucleus. So 8 nucleated. And where is it formed? It is formed in the functional megaspore. This development is called as endosporic because it is taking place within the embryo sac or nucellus. Thus, it is an endosporic development of the female gametophyte. So I hope it is clear to you. Only one megaspore participates in the formation of embryo sac, so it is called as monosporic. One at a time, only one is participating, so it is monosporic. All these terms are important. Why monosporic? Because only one megaspore participates. How it is 8 nucleated? 3 antipodals, 2 synergids, 1 egg, and 2 nuclei in the center of secondary nucleus. So 8 nucleated. But how many cells? 7 cells. Female gametophyte is formed. Now let us see what are the post-fertilization changes. In this, we'll first study about pollen-pistil interaction. It is the interaction of the pollen grain with the sporophytic tissue or the stigma. So the pollen grain, it is falling on the stigma of the flower. It begins with pollination and ends with fertilization. Pollen-pistil interaction will always begin with pollination and end with fertilization. It includes deposition of the pollen grain on the stigma to entry of pollen tube in the ovule, and it will reach the synergid. Pistil has ability to recognize, this is important, and accept compatible or right pollen of the same species. The pollen grain of the same species, if it falls on the stigma, then only the pollen tube will be formed. Otherwise, if it is of other species, it is incompatible, pollen tube formation will not take place, pollen-pistil interaction will not be successful. Only intraspecific, within the same species, pollen germination occurs. If it is to be brought about between two different species, that has to be done artificially by a process called as hybridization. But generally, only intraspecific pollen germination occurs. Pollen tube then moves because of which movement? I already told you, because of chemical movement. So these movements are called as chemotactic movement. In synthetic medium, suppose you are artificially doing all this, sucrose will induce pollen germination. You have a practical, bad luck that you are not able to perform this year. Otherwise, we give you flowers, vinca plants, and we ask you to remove the pollen grains of the vinca plant. We give you a petri dish. In the petri dish, you have to take sucrose solution, add pollen grains. When you add sucrose to these pollen grains, pollen tube germination, pollen tube formation is induced. And the tube growth will be faster if you are adding boric acid to this germinated pollen grains. So you have a practical related to it. Bad luck, you are not performing this year. But anyways, you should know that in synthetic medium, sucrose induces pollen germination and pollen tube growth will be facilitated with the help of adding boric acid to it. Artificial hybridization. Artificial hybridization is one of the major approaches used in crop improvement. You have actually one more practical of hybridization in your Balbharti textbook. You can see it, practical handbook, and it is about artificial hybridization. So it is one of the major approaches used in crop improvement. Only desired pollen grains are hand pollinated and they are used for fertilization. So this is accomplished by 3 steps: emasculation, bagging, and of course which is followed by tagging. What is emasculation? In this first picture, you can see that person is removing the anthers of the flower. So all the anthers are removed by the forceps before they are dehiscent or before they are maturing. And this process of removal of the anthers before they are antithesis takes place or before they are maturing, before the pollen grains are produced, it is called as emasculation. So if in these flowers, all the anthers are removed, all the pollen grains, all the male parts are removed, this flower is treated as a female part, correct? Then artificial cross-pollination can be brought about, and then it is covered with by a polythene bag after artificial hybridization of the required plants, species. And why is polythene bag added to it? So that covered to it, so that unnecessary pollination, unnecessary pollen grain should not deposit on the gynoecium. And a tag is added, the date on which the artificial pollination was carried out or the species which are used. All these details are written on this tag. So these are the 3 steps where artificial hybridization is done. It is accomplished by emasculation. Emasculation is removal of anthers before anthesis, before formation of pollen grains. Bagging with a polythene bag and then tagging it. So bagging and tagging. Now let us discuss about double fertilization and pollen-pistil interaction. Process of fertilization was discovered by Strasburger in 1884. After pollination, the intine of the pollen grain forms pollen tube. Through the germ pores, this is called as siphonogamy. Process when pollen tube is formed and male gametes are transferred to the ovule by a pollen tube, it is called as siphonogamy. Very important. 1 mark definition can be asked. When male gametes are transferred to the female gamete by forming a pollen tube, it is called as siphonogamy. Growth of the pollen tube is stimulated by sugary substances present on the stigma. Pollen tube with 2 male gametes, we discussed 2 male gametes runs through the style. You can see in the picture they are running through the style. and they finally turn towards micropylar end of the ovule in the cavity of the ovule. You can see they are entering from the micropylar end. Path of the entry of pollen tube in the ovule. There are three ways how it enters. If suppose the pollen tube is entering from the micropylar end, then this process it is called as sporogamy. If the pollen tube is entering from the chalazal end, then it is called as chalazogamy. Of course, it will enter from the chalazal end, again go towards the micropyle and then enter to the from the synergids. Synergids will help it. But if it is entering from the integuments, then it is called as mesogamy. So 4 definitions now you learned in last 5 minutes. Siphonogamy, process of formation of pollen tube. The male gametes are transferred to the female gamete by forming a pollen tube. It is called as siphonogamy. If this pollen tube is entering from the micropyle, we call it as sporogamy. If the pollen tube is entering from the chalazal end, we call it as chalazogamy. Whereas if the pollen tube, it is entering from the integuments, we call it as mesogamy. All these 3 definitions are very, very important. 4 definitions are important. Now let us see. This is a very nice diagram. I have taken it from NCERT books. So this is the chalazal end. At the chalazal end, these are the 3 antipodals. Functions of antipodals is not specified. Here you can see these are the 2 synergids. Okay, these are the 2 synergids, and in between the synergids you can see this is the egg. These 3 forms the eggs apparatus. In the all these are haploid. In the synergids you can see these finger-like processes which are called as filiform apparatus. In the center you can see these are the polar nuclei. Later it will fuse together to form a central cell or secondary cell which is diploid. Antipodals are haploid, synergids are haploid, egg is haploid. Only polar nuclei which fuses to form secondary nuclei later will be diploid. So style may be hollow or solid. On piercing the nucellus, the pollen tube enters the embryo sac. Tip of the pollen tube burst. When the pollen tube reaches near the synergid, near the micropyle, it burst and the 2 male gametes are discharged. The tube nucleus disorganizes and 1 male gamete fuses with the egg. This is called as syngamy or generative fertilization. Very, very important definition. When the first male gamete, it is fusing with the egg, this is first fertilization and it is called as syngamy, or it is also called as generative fertilization. The filiform apparatus. What are filiform apparatus? They are finger-like processes. Where are they formed? In the synergids. And what is that? What are they doing? They are helping the male gametes to go and fertilize the egg. Okay, to carry out syngamy. This later give rise to the embryo. What is the result of syngamy? The result of syngamy is formation of embryo. I repeat, first male gamete, when it fuses with egg, what is it called as? It is called as syngamy, or it is called as which fertilization? Generative fertilization. And what is the result of syngamy? It will give rise to embryo. Look at this picture, how the male gametes are reaching, pollen tube is reaching, how the finger-like processes are formed, filiform apparatus in the synergids, and how they are guiding the first male gamete towards the egg so that syngamy can occur, which will give rise to embryo. Now, look at this picture. So, you can see here the tube cell is coming. This is the generative cell. This is the tube nucleus. This will degenerate. The generative cell now give rise to first male gamete, second male gamete. The first male gamete is entering from the micropyle. Synergids help. It fuses with the egg and forms syngamy, which results in the first fertilization, and it will give rise to the embryo. The second male gamete, look at this picture. The second male gamete, it goes and fuses with the secondary nucleus. Now students, secondary nucleus was diploid, so it is 2n, and 1 more male gamete is fusing into it. So now what is the result? It will form a 3n, which is called as a pen. What is pen? Primary endosperm nucleus is formed. And what is the ploidy of primary endosperm nucleus? It is triploid. Why it is triploid? Because haploid male gamete is fusing with the diploid secondary nucleus. And this is called as secondary fertilization. So let us see. The second male gamete, it fuses with the secondary nucleus to form triploid primary endosperm nucleus. As there are fusion of 3 nuclei, this is also called as triple fusion. or it is called as vegetative fertilization. See, very, very important, double fertilization which is occurring in angiosperms. First fertilization, it is called as syngamy, or it is called as generative fertilization. What is the result? Formation of embryo. The second fertilization is called as the triple fusion. Result is formation of endosperm. What is the endosperm ploidy? Triple N. What is the ploidy of embryo? Diploid. What is the function of endosperm? It is going to nourish the developing embryo. This later give rise to endosperm. So triple fusion, which is vegetative fertilization, which is a fertilization of second male gamete with the secondary polar nucleus present in the center. It is called as vegetative fertilization or triple fusion, and it will give rise to endosperm. What is the function of endosperm? It will nourish the developing embryo. This process was discovered by S.G. Navashin in Fritillaria species. So this is about double fertilization. What is the significance of double fertilization? This double fertilization, it is found only in angiosperm plants. It is not there in gymnosperms. It restores diploidy. Because of syngamy, because of the fusion of first male gamete and the egg, diploid condition is restored. It facilitates further growth of embryo sac. Embryo sac further develops. Due to the triple fusion, due to the fusion of the second male gamete and the secondary nucleus, which is triple fusion, endosperm is formed, and endosperm will later provide nourishment or nutrition for the developing embryo. If fertilization fails, suppose first fertilization does not occur, then endosperm will not develop. And if endosperm is not developing, it will save energy. Thus, it avoids the wastage of energy in development of endosperm. Endosperm formation will occur only if first fertilization is successful, whereas there is no such provision in gymnosperms. Very important, it also helps to avoid polyembryony. Polyembryony condition is a condition where many embryos are formed from the nucellus. And because of double fertilization, where only two male gametes are formed, many embryos are not formed in most of the angiosperms. Of course, there are polyembryony in some of the angiosperms. We will see about it later. So, significance of double fertilization found in only angiosperms, restores diploidy, further growth of the embryo sac will be continued. Because of triple fusion, endosperm is formed. Endosperm will provide nourishment to the developing embryo. If fertilization does not occur, it will not form the endosperm, thus serve the energy. And no such condition is there in gymnosperms, and it also helps to avoid polyembryony. Yes, students. So we studied many important concepts today. First, we studied the structure of Anatropus ovule, how it is bitegmic, chalazal end, micropylar end. Then development of the female gametophyte from a single sporogenous tissue present in the nucellus, which give rise to 8 nucleated, 7-celled female gametophyte. And then we also studied how double fertilization takes place, which consists of syngamy, resulting of formation of embryo, and the triple fusion, which is resulting in formation of endosperm. So I hope you have understood all these concepts. We'll continue next time. Thank you.